Hi everybody, this is Mrs. Mancuso, and this AP Bio video is about the signal transduction pathway. So, first thing I want to talk about is here's a cell, and here's some hormones, and hormones signal cells. So this video is all about how do signaling molecules work, and how do they cause a change inside of a cell. So the first thing that we know is there has to be receptors. There has to be something on the cell that's very specific that can actually receive and match that hormone. And it doesn't take the hormone into the cell usually, but it causes a change. And it goes through a pathway, and it will induce other molecules to be produced and maybe released outside of the cell. And so there's 3 steps here. The first step here is reception. The next step is transduction. And the last step is response. So you need all 3 of these steps in order to actually have a reaction. So I have a little story. Here's Spencerport High School. And Spencerport High School, I mean, we are known for our super school spirit, and we'll do just about anything. We're up for crazy challenges. So here comes this man, and he comes up to the school, and he's greeted by Mr. Olson. And Mr. Olson says, "Oh, you can't enter here." And he says, "Well, I've got a challenge for the school. I want to know if you guys can break the world record and make the most paper airplanes and shoot them out of the school all at once." And he says, all right, I will pass on this message to the school, and we'll see what we can do. So the man walks away, and Mr. Olson goes into the main office. And when he goes in the main office, he talks to Mrs. Adamick and talks to Miss Hill and talks to everybody, and goes and finds Mr. McCabe and sort of begins the transduction step, which means that he is talking to people and spreading the message. Well, you all know Miss Adamick gets right on that PA system and starts giving us notifications and messages. So she starts telling the whole school. And each room starts making paper airplanes and getting all ready. And eventually the next day we decide, hey, let's do this. And we've made all these paper airplanes, and we let them all go. So all of those were actually steps. This guy here, the man that came to the front door but didn't actually enter, he is the signaling molecule. We call signaling molecules ligands, and the most common ligands are hormones and neurotransmitters. And again, usually hormones and neurotransmitters don't actually enter a cell, but they do cause a change. So the next thing we had was Mr. Olson right here. And Mr. Olson was our receptor. He received the message, and he carried that message into the cell. And then within the cell, we had multiple molecules that were getting turned on. And all of this makes up the transduction step. So transduction is the spreading of a message, and it usually follows what's called a phosphorylation cascade. So I'm going to write this word here because it's very important, and I want you to know what it means. Phosphorylation cascade. So to phosphorylate means to add an inorganic phosphate to something. And if you have an inactive enzyme and you add a phosphate group to it, it becomes an active enzyme. And an active enzyme is going to go ahead and start working. So it was off, and now this is a signal molecule that is turning it on. Now inside of a cell, you can imagine, here's my receptor, here's my ligand, and this is right in the membrane. And inside of here is one of those molecules that is going to begin the phosphorylation cascade. So this is an inactive enzyme. And when this receptor receives the ligand here, it actually changes shape, and it releases molecules that regulate this structure. And this structure then becomes phosphorylated, so you can think of this as the passing on of phosphates. And as it becomes active, it starts to work. And so let's say it starts making all of these molecules. It doesn't just make one, it makes many. So this process is definitely known for amplification. To amplify means to increase a signal. So we only had one ligand out here, but we could have 1,000 or more molecules made inside of the cell because of that one signal. So let me give you another example. Here's a cell, and here's that same receptor. This time the molecule right here is adrenaline. So think about adrenaline. Adrenaline is a hormone that your body makes when you're responding to a stressful situation. So you're bringing out the garbage in the morning and there's a bear. You don't usually see bear in Spencerport, so this is like a pretty freaky thing. So your body responds with what's called the fight or flight response, and you're going to react. You are either going to fight that bear or you're going to run away. I'm guessing we're going to run. Well, to run, your body floods glucose. So inside of a cell, so here's inside my cell, I don't have glucose, I have glycogen. So glycogen is my stored polysaccharide. So here's a big long glycogen molecule. And once the cell receives adrenaline, I've got to turn on a transduction pathway in order to break up this glycogen and make glucose, because I've got to respond to the bear and I need to run away fast. So this is a really, really fast process in cells. So what happens is this signal molecule, it actually has what's called a G protein on it. And the G protein undergoes a conformational change. So that means it changes shape. And it activates other enzymes that are right in the membrane. So in the membrane, this is adenylyl cyclase. And adenylyl cyclase ends in ACE, so it's definitely an enzyme. And it makes cyclic AMP. So you know ATP is adenosine triphosphate. AMP is adenosine monophosphate. So it only has one phosphate on it, and cyclic means it actually goes around in a circle. So the molecule circles up with itself. So it starts producing this cyclic AMP. And cyclic AMP is part of that phosphorylation cascade. So it activates enzymes within the cell. Well, the enzyme that we really want to activate, well, first of all, I'm getting a little bit ahead of myself. The enzymes, as they are activated, are called kinases. So kinases are a great example of an inactive enzyme that will be activated by cyclic AMP. So this turns this on. Now, kinase is a regulatory molecule. Again, ACE at the end, so it's an enzyme. Kinase will turn on a very specific enzyme. There are a lot of kinases, so they'll turn on a lot of things. But in this situation, it turns on glycogen phosphorylase. You'll notice it seems like these words are like huge and overwhelming, but they're really not. Glycogen is the polysaccharide. Phosphorylase means that when it has a phosphate group attached to it, and ACE means it's an enzyme. This glycogen phosphorylase, this is the enzyme that will change glycogen into glucose. So it does hydrolysis. It breaks the bonds. So if we go back here to my picture of the cell, I've got the signal molecule, that's adrenaline. And then here, this was my receptor. It signaled adenylyl cyclase to make cyclic AMP. That activated some kinases. The kinases activated this enzyme, glycogen phosphorylase, which cuts apart glycogen. If you cut apart glycogen, you get glucose. It's now going to be in very high concentration, and it's going to move on out. Where does it go? Into your blood. What do you need if you see a bear when you're bringing out the garbage? You need sugar in your blood so that you have sugar going to your muscle cells and you can run away. So all of that happens in such a quick time, but it's a perfect example of the signal transduction pathway. So again, because I just want to make sure that you have these steps down. Step 1 is always reception. Reception means you got to receive the message. You're relying on two things. You're relying on a ligand, that's the signal molecule, and you're relying on a receptor. These have to be a match. If they're not a match, it will not signal it and it will not change. You have a lot of cells in your body, and ligands only work on target cells. So your body might make adrenaline, but it only targets specific cells that actually will change. Those target cells will have the right receptors. All right, the second step in the pathway is transduction. Transduction is how you spread the message. You spread the message by intermediate molecules. You usually have a secondary messenger like cyclic AMP, and you have kinases which change from inactive to active. When they are active, they induce the phosphorylation cascade. And again, that means you are activating enzymes by adding a phosphate group. All right, the last step of our signal transduction pathway is always response. So a response is your end result. You got to have something happen because of this pathway. So let me give you one more example. Here's my cell, here's a receptor, and here's a different signaling molecule. I should make my receptor look like it completely receives it. All right, there we go. Now, inside of here, I have the nucleus, and inside the nucleus, I have DNA, and I have tons of DNA. So you know that you have 46 chromosomes, 23 different pairs. Each one of those chromosomes can have anywhere from 100 to 1,000 genes on it. And each one of the genes is responsible for building a specific protein. You don't build all of your proteins all the time. There's no need to. So instead, you need what are called transcription factors. So transcription is the process of making RNA from DNA. And a transcription factor initiates transcription. So I am going to have my signaling molecule right here. And this is step 1. There's reception. Step 2 is my cascade. So to activating kinases, making things. And then the next thing I need is I need a transcription factor to be activated. So this is going to be a transcription factor, and it actually binds to the DNA. When a transcription factor binds to DNA, it causes RNA to be made. RNA goes out of the nucleus because it's small, and RNA goes to a ribosome, and it's going to build a protein. So this protein right here, that's step 3. That was made in response to this pathway. If there hadn't been step 1, there would not have been any need for 3 to be made. So this protein here, this squiggle that I made, that would not have occurred without this signal molecule. So step 1, I had to have reception. Step 2, I had to have this pathway that produced transcription factors that turned on a gene and made RNA. And then the RNA went out to the ribosome and actually built a protein. So that end result is only made when it's signaled. Your body will not go through that process unless it is necessary.